Hello everyone, my name is Snehlata and I'm a PhD student and a PMR fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today, we will continue with the next lecture of the chapter Photosynthesis in Higher Plants. So, the last lecture we have completed about the light reaction of the photosynthesis. Right? The first reaction is light reaction that we saw in the previous class. So the first reaction is light reaction, and the products that are formed during the light reactions were, so the products were ATP, NADPH, and oxygen. And this oxygen is what? This is byproduct. Right? Now, the second part of this photosynthesis reaction is dark reaction. So, the second part is dark reaction. And now, what does dark reaction have? So, the dark reaction, it is known as biosynthetic step. Okay, the dark reaction of photosynthesis is known as biosynthetic phase, or you can say step. While this light reaction is known as photochemical reaction, because here the light is used as energy, right, to convert the substrate into certain kind of chemicals. So it is known as photochemical phase or step. Okay. Now, this dark reaction is known as biosynthetic phase. And what happens in the dark reaction? In the dark reaction, fixation of CO2 takes place. So, fixation of CO2, and by fixation, what does it mean? It means that conversion of CO2 in some organic molecules. So, conversion of CO2 in organic molecule. Now, what this organic molecule can be? So this is what? Glucose. But we have studied in the first lecture that if you look into the chloroplast, because we know that whole this reaction takes place in the chloroplast, right? So if you look into the chloroplast and try to find the first product of the photosynthesis, you will not find glucose there. Why? Because this glucose, it gets converted into starch. And this starch, it is stored. Okay. So in the dark reaction, what happens? It is the phase where the fixation of CO2, which means the conversion of CO2 into organic molecule, takes place. And the glucose that is formed, that is either stored in the starch or it is transported to other path. Okay. So it is either stored or it is transported to other part. So this is what happens in the dark reaction. Now, the dark reaction, it is, and now we have also understood that why this is called a biosynthetic phase, because what happens here, the synthesis of organic molecules takes place. So it is known as biosynthetic phase because of synthesis of organic molecules takes place in this phase. Okay. Now this dark reaction, it comprises of, just like light reaction, it has like this cyclic photophosphorylation and non-cyclic photophosphorylation. Similarly, the dark reactions also comprises of different type of cycles. Okay. So the dark reaction consists of different cycles. Okay. Consists of different cycles, and what is the basis of these cycles? So these cycles takes place in different type of plants. Okay. So if you we do not we categorize the plants based on the type of dark reaction in which that takes place in that particular plant. Okay. For example, you have the first cycle is known as C3 cycle. The first cycle is known as C3 cycle. The second common cycle is the C4 cycle. And the third one is the C2 cycle. Okay. Now, what is the meaning of this C3, C4, C2? So what does this number means? So this number means, or this cycle is named based on the first stable product that is formed in that cycle. Okay. So if a product that is formed is of 3 carbon molecule, then it will be called as C3 cycle. Similarly, if it is formed, the first stable product that is formed is of 4 carbon molecule, then it will be called as C4 cycle, and likewise. Okay. So these cycles are named. The cycles are named based on the first stable product formed, based on the number of molecules in the first stable product that is formed. Okay. Number of carbon molecules. Okay. Number of carbon molecules in the first stable product that is formed. First stable product formed. Okay. So what happens when this cycle starts? The first stable product that is formed, they count the number of carbon in that molecule, and based on that, this cycle name was given. Okay. For example, in C3 cycle, first stable product is of 3 carbon molecule. Okay. First stable product is 3 carbon. I'm representing 3 carbon by 3 C. So 3 carbon molecule. Okay. So this is about the dark reaction. Now we will study these cycles one by one. So the first cycle is C3 cycle. Okay. And C3 cycle, it is also known as Calvin cycle. It is also known as Calvin cycle. Now, why it is known as Calvin cycle? Because it was discovered by a scientist named Calvin. Okay. Discovered by Melvin Calvin. Okay. So this is the name of the scientist. So discovered by Melvin Calvin, and that's why it is called as Calvin cycle. And in which organism this cycle was discovered? So it was discovered in algae, the green algae, that is chlorella. That is chlorella. Okay. So chlorella is the green algae in which the Melvin Calvin first time discovered the C3 cycle or the Calvin cycle. Okay. Now, how does he discover this? So he used a technique known as radio label or radioisotope labeling. Okay. So he used technique known as radioisotope labeling. Radioisotope labeling. So what happened in this radioisotope labeling? So in the radioisotope labeling, instead of using a general element, suppose carbon. So we know that carbon has different isotopes. So carbon 12, carbon 13, carbon 14. So instead of using carbon 12, the scientists can use another carbon isotope, say carbon 13 or carbon 14. Okay. So when they will analyze these things when they will analyze the products that are formed, they can trace those carbon molecules based on the radioactivity. And based on that, they can determine that how many number of carbon molecules are present in that product. Okay. So what means? It means that instead of, most naturally occurring carbon, they use carbon radio as another isotope of carbon. So used different isotope of carbon. And then they can easily analyze the number of carbon molecules that are present in that product by this radioactivity. Okay. Now, the C3 cycle is known as universal cycle. Okay. So C3 cycle is known as universal cycle. Now, why it is known as universal cycle? So first, this is known as universal cycle. Okay. Now, as the name suggests, universal means present in all kind of plants. So whatever plant is there, this C3 cycle will definitely happen. Okay. But in previous this slide, what I told that these dark reactions consist of different cycles, and these different cycles happen in different plants, right? So if a C3 cycle is happening somewhere, that plant will be called as C3 plant. If C4 is happening, that plant will be called as C4 plant. But here I am saying that the C3 cycle is known as universal cycle, which means that this cycle is occurring in all kind of plants. So how is it? How it can happen? So what happens that although different plants have different cycle, for example, in C4 plants, it has C3 cycle, but at some point of time, that C4 cycle will get converted into C3 cycle, or that C3 cycle will be responsible for the C4 cycle. So something like that will be happen that we will see in upcoming slides. Okay. So irrespective of type of plant, the C3 cycle is a universal cycle. So if someone asks that if you have a C4 plant, so does that what kind of cycle it will have? So it will have both C3 cycle as well as C4 cycle. So this is what is meant by a universal cycle. Okay. Now, what is the location? So what is the location of C3 cycle? Okay. So the C3 cycle, we know till now we have understood that everything that we talk about was happening in the chloroplast, right? In the leaves, we have mesophyll cells, and in the mesophyll cells, we have chloroplast, and everything was happening there. So, and we know that this light reaction, it takes place on the thylakoid membrane. But where does C3 cycle happen or the dark reaction happen? So it takes place in the stroma. So C3 cycle, so if this is your chloroplast, this is your thylakoids, and this part is stroma. So C3 cycle will happen here. Okay, this is the location of the C3 cycle, this stroma. Okay. Now, what is the enzyme? Because in previous lectures, we have seen that the dark phase is known as biosynthetic phase, and it consists of enzymatic reactions. So there must be some enzyme that is present that catalyzes the reaction, right? So the enzyme that is present or that is used for C3 cycle is known as rubisco. The enzyme is known as rubisco. Okay. Now this rubisco, this is the most abundant enzyme in the biosphere. It is the most abundant enzyme in entire biosphere. In entire biosphere. Okay. So this is the most abundant enzyme in the entire biosphere. And if you talk about the living organism, or you can say if you talk about the human body. So, okay. So if you talk about the earth, then the most abundant this enzyme is cellulase. Okay. Now, this enzyme, or you can also, the question can also be in terms of proteins, because we know enzymes are what? Mostly proteins. So rubisco is the most abundant enzyme in the entire biosphere. Okay. Now, what does this rubisco stands for? So the rubisco, it stands for ribulose. The full form of rubisco is ribulose 1,5-bisphosphate. Bisphosphate means that on ribulose, we have phosphate at first and fifth position, right? Bisphosphate, the C stands for carboxylase, and O stands for oxygenase. So the enzyme rubisco, it stands for ribulose 1,5-bisphosphate carboxylase oxygenase. Okay. So as the name suggests, it comprises of two terms, carboxylase and oxygenase, and that makes this enzyme bifunctional. So ribosco is what? It is bifunctional. Okay. Now it has two functions. The first is it acts as a carboxylase. Okay, act as carboxylase. Now, what happens, or under what condition it acts as carboxylase? So, it acts as carboxylase when on the active site of this enzyme, because this is an enzyme, it will have active sites, right? So, on the active site of the enzyme, carbon dioxide binds. Okay, carbon dioxide binds, and the second substrate of Ribisco is RUBP, about which we will see in upcoming slide. So, this Ribisco, it acts as a carboxylase where on one active site the carbon dioxide binds. Okay, and this happens in the case of C3 cycle. Now, there can be another situation where it acts as oxygenase, where it acts as oxygenase. And what happens in this case? In this case, in its active site, instead of carbon dioxide, the molecule that is binding is oxygen. And the second substrate will be RUBP. Okay. And this type of cycle is known as C2 cycle. And the plant don't prefer this thing, that Ribisco acts as oxygenase, but there are certain conditions in which it has to act as oxygenase. Okay. So this is the brief introduction about the C3 cycle, where it takes place and what is the enzyme involved. Now, if you look at the C3 cycle, so as we know, it's a cycle. So it will work like this, right? It will start from here and then it will come back here. So in that cycle, it consists of 3 steps. And the first step is carboxylation. The first step of C3 cycle is carboxylation. So what happens in carboxylation? So here what we are doing, we are fixing carbon dioxide. So definitely it will need carbon dioxide. So in carboxylation, carbon dioxide will come. Okay. And then the second substrate is known as RUBP that I was saying. So this RUBP, it stands for ribulose 1,5 bisphosphate. Bisphosphate. Okay. And it's ribulose, so it's a 5 carbon molecule. Okay. So it is what? 5 carbon. This is 1 carbon. And the structure of RUBP will be, let's draw, it has 5 carbon. So 3, 4, 5. Okay. And then you have CS2. What it is saying, that it has phosphate as position 1 and position 5. So we draw position at position 1 and at position 5. We have added this phosphate. And then it's a ribulose. So it will have ketone group. And then it will have CS2, CS2. So this is the structure of ribulose 1,5 bisphosphate. And it is 5 carbon molecule, right? And the CO2 is 1 carbon molecule. Now, in the presence of this enzyme, that is Ribisco, what happens? It gets converted into a 3 carbon molecule. Okay. So this is 1 carbon, this is 5 carbon, but they are getting converted into 3 carbon molecule. Now the question is, the total carbon that we have is 6. And if it is getting converted into 3 carbon molecule, then how many molecules of that 3 carbon substrate product will be formed? So it will be 2, right? So it will lead to the formation of 2 molecule of, and the product that is formed is known as 3-phosphoglycerate, 3-PGA. Okay, and this PGA stands for phosphoglycerate. Phosphoglycerate. Okay, so the name of the product is 3-phosphoglycerate, and this product is of 3 carbon. So this is 3 carbon molecule. 3 carbon molecule. Okay, so what has happened in carboxylation step? In carboxylation step, the addition of carboxyl group to RUBP takes place, and this leads to the formation of 2 molecule of 3-PGA. 3-PGA is the product name, and it is of 3 carbon. Okay. Now, so here what we have done, we have fixed 1 carbon, that is 1 CO2. But we know the formula of glucose, it is C6H12O6. So it comprises of 6 carbons, right? So in order to fix or produce 1 glucose molecule, how many molecule of carbon will be needed? So we will need 6 carbon of molecule, 6 molecule of carbon dioxide. So we need 6, or we can write for one glucose molecule. So for one glucose molecule, what do we need? We need 6 carbon dioxide, 6 molecule of carbon dioxide plus 6 RUBP. Now, if there are 6 molecule of carbon dioxide and 6 molecule of RUBP, how many molecules of PGA, 3 PGA will be formed? So one molecule leads to 2 molecule formation of 3 PGA. So 6 carbon molecule will lead to 12 molecule formation, right? So 12 molecules of 3 PGA will be formed. Okay. So this is the carboxylation step. Now, the next step is reduction step. Okay. So it comprises of 3 steps. First is the carboxylation. The second step is the reduction step. Now, what happens in the reduction step? So in the reduction, now what substrate we have? We have 12 molecules of 3 PGA. 12 molecules of 3 PGA, 3-phosphoglycerate. Now, as the name says, it is a reduction. So this PGA will undergo reduction. Okay. And once it will go reduction, it will lead to the formation of 12 molecules of 1,3-BPGA. BPGA. Okay. 1,3-BPGA. And this is again 3 carbon. Okay. This is also of 3 carbon. And BPGA stands for 1,3-bisphosphoglycerate. Bisphosphoglycerate, or you can also say bisphosphoglyceric acid. Okay, glycerate or glyceric acid. Okay, so this is the reduction step where 12 molecules of 3 PGA gets converted into 12 molecules of 1,3-bisphosphoglycerate. Now, what is the catch here? So when we were studying about the dark reaction, we said that the dark reactions are those that do not happen in the dark, but they do not directly depend on the light energy. That's why they are called as dark reaction, right? But what this dark reaction do? They uses the product that is formed in the light reaction. Now, what were those products? Those were ATP and NADPH. So where these products will be used? The product will be used here. Okay. So in the reduction of 12 molecules of 3 PGA to 12 molecules of 1,3-BPGA, 12 molecules of ATP will be used. Okay. So this is formed during the light reaction. Now it is getting used here. So 12 molecules of ATP is used to convert 3 PGA to 1,3-bisphosphoglycerate. Now this 12 molecules of 1,3-BPGA again undergo reduction. Okay. So this 12 molecules of 1,3-BPGA, they again undergo reduction to form 12 molecules of 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde. Okay. Now here what you had glycerate, or you can say glyceric acid. So here you had acid, here you have aldehyde. This is what? This is reduction, right? You have COOH and it's getting converted into CHO. You have removal of one oxygen, right? So it is reduction. So what is happening? The reduction is taking place. And what is being utilized here? Here, the another molecule or another product of light reaction is being used, that is NADPH. So here 12 NADPH molecule is used. Okay. So this is the reduction process where what happens? 3 PGA first gets converted into 1,3-BPGA, and this 1,3-BPGA gets converted into 3-phosphoglycerate. And this leads to the utilization of 12 ATP and 12 NADPH. Now, the last step of the C3 cycle is known as regeneration step. Now, what happens in regeneration? We have 3 molecules of phosphoglyceraldehyde that we can also mention as 3-PG. Okay. So this 3-PG, we have 12 molecules of 3-PG. Now, what happens out of this? Because this 3-PG is of 3-carbon molecule, right? It is 3-carbon containing molecule. Now we want to produce glucose from here. So to produce glucose, how many molecules of 3-PG will be used? Since one molecule consists of 3 carbons, we need total of 6 carbons to produce one molecule of glucose, right? So out of these 12 molecules, 2 molecules will be used. 2 molecules will be used to produce glucose, right? Because one molecule have 3 carbons, 2 will be have 6 carbons, and it will lead to the production of glucose. And the remaining 10 molecules, what happens to them? So these 10 molecules, they are used to regenerate RUBP. They are used to regenerate RUBP. So this is what happens. Okay. Because if you see the reaction, the RUBP is also being utilized. And this RUBP has to be recovered or regenerated, right? So this is regenerated by this process. Okay. So this is the complete C3 cycle. Now let's see it in the form of cycle, because this is a cycle. So what is happening here is, you can see all the 3 steps I mentioned. So the first here is your, from here till here is your carboxylation. From here to here, this is your reduction and then regeneration. So this is a carboxylation, this is your reduction, and this third is your regeneration. Okay. And now what we are seeing, that here ribulose 1,5 bisphosphate plus CO2, it undergo carboxylation, leading to the formation of 3-phosphoglycerate. So what is 3-phosphoglycerate? This is the first product, first stable product of C3 cycle, right? And this is what? This is 3 carbon. This is 3 carbon. And that's why it is called as C3 cycle, right? Now, the another thing that we are seeing is, in the reduction step, we have ATP and NADPH being used. So this cycle is in terms of one carbon dioxide molecule. But here we know that in the first reaction itself, here the 2 molecules of this 3-PGA forms, right? So the ATP and NADPH that will be used during one cycle is 2 ATP and 2 NADPH here. Okay. So in one cycle, 2 ATP and 2 NADPH will be used. And this will lead to production of 2 ADP and 2 NADP plus. And in regeneration, one molecule of ATP is being used. Okay. So this is a C3 cycle. Now, this complete cycle was in terms of one carbon dioxide fixation. But if we talk about the one molecule of glucose, so for one molecule of glucose, what happens? How many ATP and how many NADPH and how many turns of C3 cycle will be required? So for one molecule of glucose, we need 6 turns of C3 cycle, right? 6 turns of C3 cycle, because in one turn it is fixing one carbon dioxide, but we need a glucose that is of 6 carbon. And because of this reason, we need 6 turns of C3 cycle. Okay. Now, how many ATPs are required for one molecule of glucose formation? So we can see that in one cycle, we can write here, in one turn, how many ATP and how many NADPH is required? Here is 2 ATP and here is 1 ATP, right? So in one turn, 3 ATP plus how many NADPH we have? We have 2 NADPH, right? Plus 2 NADPH is required. NADPH is required, right? So for 6 turns or one glucose molecule or one glucose, we need 18 ATP and 12 NADPH, right? So for one turn, we need 3 ATP and 2 NADPH. Then for 6 turn or for one glucose molecule, we need 18 ATP and 12 NADPH, right? Now, this is one thing. Now, the second thing, let's see. If someone asks that in the case of reduction only, so in reduction step, what happens? So someone asks that in reduction step, for one turn, how many ATP and NADPH is required? So in reduction, for one turn or for one carbon dioxide fixation or one CO2 fixation, the number of ATP and NADPH required is 2 ATP. We can see from the figure, right? 2 ATP plus 2 NADPH, right? Then for one glucose or for 6 turn or 6 CO2 or one glucose, how many ATP and NADPH is required in the reduction step? Okay, we need 12 ATP and 12 ATP and 12 NADPH, right? So this calculation is important. 12 ATP and 12 NADPH. Okay. So this is the reduction step. And similarly, how the number of ATP that is formed, it will lead to the release of that much of ADP. So if 12 ATP is formed, then 12 ADP will release, right? Similarly, if 12 NADPH is used, then 12 NADP plus will be released. So it will work like this. Okay. Now the third step is what we have, regeneration. So for regeneration, if somebody asks that how many molecules of ATP and NADPH is required for one turn, so for one turn, we can see 1 ATP is required and 0 NADPH. So it means that in regeneration phase, we do not need NADPH, right? So regeneration do not require NADPH. Okay. Now, so for 6 term or a glucose, we need to utilize 12 ATP in regeneration phase, right? Sorry, 6 ATP, because it will take 6 turns, right? So we need 6 ATP. 6 ATP for in the regeneration phase. Okay, so this is how the calculation is happening. For complete cycle, this is a calculation. So this is for complete cycle. This is for reduction step and the third is for regeneration step. Now, there are a few questions. For example, what is the primary acceptor of carbon dioxide? So, primary CO2 acceptor. What do we have? So the primary acceptor of CO2 is RUBP, right? This RUBP. So it is RUBP. Okay, so RUBP is a substrate and RUBP, Rubisco, is the your enzyme, right? Now this RUBP is of 5 carbon. Okay, and the first stable product that is formed is 3-phosphoglycerate. And which is the most important step of C3 cycle? So the most important step of C3 cycle, it is carboxylation. Okay, so the most important step is carboxylation. Okay, so with this we complete our C3 cycle. Now, what we saw when we were studying about Rubisco, we saw that the Rubisco act as both the carboxylase and oxygenase, right? So when it acts as carboxylase, we saw what happened. The C3 cycle happened, right? But what happens when it acts as oxygenase? So in that case, photorespiration happens. Okay. In this case, photorespiration takes place. So what happens in photorespiration? First of all, here you have this ribisco enzyme, right? So this is its active site because enzymes are active site. So this is its active site. And on the active site, either CO2 or oxygen can bind. This happens, right? So this ribisco, it acts as oxygenase and carboxylase, both. Right? Now, at the for its active site binding, this carbon dioxide and oxygen both compete. So CO2 and oxygen compete with each other for binding to ribisco active site, right? For binding to active site of ribisco. So this CO2 and both compete. So what kind of inhibition this will be? So this will be called as competitive inhibition. So in ribisco, what kind of inhibition takes place? Competitive inhibition, because at the active site, two substrate compete to bind. Okay, so this is known as competitive inhibition. Okay, now under what condition this ribisco will act as? So this is your ribisco. Under what condition the ribisco will act as oxygenase, and under what condition it will act as carboxylase? Okay, so the first condition is when carbon dioxide concentration is high. When carbon dioxide concentration is either high or equal to oxygen concentration. Okay, so if the concentration is either high or equal to oxygen concentration, what happens? Ribisco act as carboxylase. Okay, act as carboxylase. Which it means what? It means if you talk about the affinity of ribisco, you can see that even if the carbon dioxide concentration is equal to oxygen concentration, then also the ribisco is acting as carboxylase, right? It means this ribisco has high affinity for carbon dioxide, right? High affinity for carbon dioxide. Okay. So it means here that when carbon dioxide concentration is high, it will act as carboxylase, or even if it is equal. But in the second condition, when the condition is reversed, that is when oxygen concentration is greater than CO2 concentration. Okay. Not equal, I'm saying greater than. Okay. Then it acts as oxygenase. Okay. Then it acts as oxygenase. Now, what happens when it acts as oxygenase? So this is your ribisco, right? This is your ribisco. Okay. We know at one side it will have RUBP, that is its ultimate substrate. And then at second place, now when CO2 concentration is high, when O2 concentration is high, O2 will come and bind here. Now what happens? It will lead to the formation of two type of products. The first product is PGA, 3-PGA. The first product is 3-PGA that we have also seen in the Kelvin cycle, that is 3 carbon. Okay. And the second product is what? Phosphoglycerate. Phosphoglycolate. The second product is phosphoglycolate. Okay. And this is what? This is 2 carbon. And this phosphoglycolate, it will ultimately undergo multiple steps processing, and then finally it will release carbon dioxide. It will release carbon dioxide. Now we can see the problem here, right? So the carbon dioxide that was meant to be fixed, that was meant to be taken, it got released, right? So this is not right, because the plant need to fix carbon dioxide, not to release them. The plant release oxygen. So here, this what happens if there is no carbon dioxide? It will lead to no synthesis of any organic compound, right? So no organic compound will be formed. No organic compound will be formed, right? Because CO2 is released. And because of this, the plant do not need this process. This is complete waste process. So photorespiration is what? It is wasteful process. Okay. Here what is happening? And it is respiration. Why it's named as respiration? Because what we are seeing is it is taking, it is releasing what? CO2. And this is what happens in the respiration, right? We release CO2. Okay. So, and what it is taking oxygen here. The Revesco is binding with the oxygen. So it is taking oxygen and releasing CO2. Because of this process is known as respiration. And it is happening in during photosynthesis. That's why it is known as photorespiration. Okay. So now we saw that it is known as wasteful process. So why it is called as wasteful process? So the first we have seen because here no organic compound is formed. No organic compound is formed, right? No organic compound is formed. Now, second thing is what happened that rather than CO2 fixation, CO2 is getting released, right? So rather than CO2 fixation, it gets released. It gets released. And the third point is here it uses ATP. Okay, this process also it uses ATP. So again, it is utilizing energy, but it's of no use. That's why it's a wasteful process. Okay. Now, the photorespiration, it does not happen in only one organ. Okay. So for the photorespiration to take place, it should have three organelles. Okay. So the organelles that participate in photorespiration participate in photorespiration. So the organelles are what? CPM. CPM. Okay. And like I said in the previous thing, that this phosphoglycolate that is formed, it undergo multiple steps, right? And then finally release CO2. So these steps takes place in different organelles. Okay. So now what does CPM stands for? So the C here stands for chloroplast. C stands for chloroplast. So what happens in the chloroplast? Glycolate synthesis takes place. Okay. This phosphoglycolate. So glycolate synthesis takes place. Chloroplast, we have seen glycolate synthesis takes place. Okay. Now, the second organelle is stands for P. That starts with P is peroxisome. Peroxisome. Now, what does peroxisome do? So this glycolate, when it reaches the peroxisome, it undergo certain modifications and leads to glycine synthesis. The glycine is an amino acid, so it leads to glycine synthesis, and then finally it reaches to mitochondria. Okay, it reaches to mitochondria, and here carbon dioxide release takes place. Release takes place. Okay, so we can see the glycolate synthesis by utilizing oxygen by Revesco. It takes place in the chloroplast. Then this glycolate moves to the peroxisome where it gets converted into glycine. And then it, by certain chemical reactions in the mitochondria, leads to the carbon dioxide release. Okay. Now, and all these reactions, all these three reactions that we are talking about, these are light dependent reactions. Okay. These are light dependent reactions. Okay. And they require ATP. Okay. Because we can see here ATP is required, but nothing is formed. Okay. Now, The question is, if this is such a wasteful process, then why plant does this process, right? Or under what condition photorespiration takes place? So here we can write here. So conditions for photorespiration. Conditions for photorespiration. Okay. So what are the conditions? The first condition is, we know that when we have high oxygen concentration, when like trees like canopies, say here. So what happens? They have very much oxygen here. Okay. So in these conditions, when there is high oxygen concentration, then photorespiration takes place. Second is when there is high light intensity. High light intensity. Okay. Third condition is when there is high light intensity. You can also remember it as high temperature. High temperature. And the fourth is, low carbon dioxide. Of course. Okay. So under these conditions, photorespiration happens. Okay. Now, where does it happen? So all these conditions where they will find, we can find them most commonly. So the conditions where this happen is they occur in tropical regions. So these occur in tropical regions near equator. Regions near equator. Okay. So because we know the equator, the sunlight falls directly. So there will be high temperature, high light intensity, right? So it mostly occurs in the plants that present in the tropical region near equator. Okay. So this is about the photorespiration. So with this, we have completed the C3 cycle and photorespiration. Now we will see about another cycle, C4 cycle. But before that, let's see C4 plants. So what are C4 plants? So the C4 plants, they are found in dry tropical region. Okay. So where C4 plants are found? They are found in dry tropical region. Okay. Dry tropical region. Okay. Now, the plant population that comes under C4 plants are only 1%. Okay. So it comprises only 1% of total plant population. Okay. So C4 plants are very small in number and it comprises of only 1% of total plant population. But these plants are most productive plants. So out of all the plants, these are most productive plants. Okay. And these plants, so these comes under both monocots and dicots. So they consist of, C4 plants consist of both monocots and dicots. Okay. And the examples are important. So the examples of these plants are maize. Okay. We have sugarcane. We have sorghum. So all of these comes under C4 plants. Okay. One of the important feature or the most important feature of the C4 plants are they do not show photorespiration. So they do not show photorespiration. So does it mean that they do not have rubisco? The answer is no. Okay. So these plants also consist of rubisco, but they do not show this photorespiration. Why this is the case, we will see. Okay. Now, the C4 plants, their leaves have a special anatomy. Okay, their leaves have special type of anatomy. So the leaves of C4 plants have special anatomy. Have special anatomy, and this anatomy is known as Krenz anatomy. Okay, this is known as Krenz anatomy. Now, what is this Krenz anatomy? So this Krenz anatomy, this is what, if you see it under the microscope, you would take a leaf and dissect it and visualize it under the microscope. You will see crown or concentric ring-like structure. Crown or concentric ring-like structure. Okay. So because of this type of anatomy is known as Krenz anatomy. Now, what we will see in the microscope, let's see. So we know in the leaves, we have this vascular bundle. In plants, we do have vascular bundles, right? Now, this is a vascular bundle. You will visualize it. Now, after this vascular bundle, the second thing that you will see will have a structure something like this. A very compact structure. Okay, so these are basically cells, but these are very compact cells. Okay, and they have multiple layers. They have multiple layers. Okay, so very compact and multiple layers. And then you will see some other arrangement that will be like this. Okay, that will be like this, surrounding it. So they are close by, but not very compact. So what is this structure? Okay, so this is a leaf, and this kind of structure you are seeing. Now, what do we have here is this concentric structure that we are seeing is this thing, the very compact one. These are the special type of cells known as bundle sheath cells. These are bundle sheath cells. Okay, and these bundle sheath cells, these are what? These are compact. These are compact and multiple layers and have multiple layers. Okay, so this is your bundle sheath cells that are located outside this vascular bundles. Now we know that the photosynthesis takes place in mesophyll cells, right? So does the C4 plants do not have mesophyll cells? The answer is no. If you see here, this green cells, these are what? These are your mesophyll cells. Okay. So now you have seen that this leaf anatomy, this is the leaf anatomy. Okay. So what we see that along with the mesophyll cells, these have also another type of cell that is known as bundle sheath cells. Those are and these are compact in nature. Now let's see what is the difference between this mesophyll cells and bundle sheath cells that is present in the C4 plants. So first feature differentiation is based on the size. So if we talk about the size, these mesophyll cells, these are small in size, while these bundle sheath cells are large. Okay. Now, what kind of cell wall do they have? So the mesophyll cells, they have this thin and gas permeable cell wall. Okay, so it allows the gas to pass through it. While this bundle sheath cells these are thick and permeable to almost everything. Okay, so these are thick and permeable. Now, if we talk about the arrangement of this mesophyll cells, so this mesophyll cells arrangement is relatively loose. Okay, so they are like, say, this, so they are in loose contact with each other, while the bundle sheath cells, they have compact arrangement. They have compact arrangement. Now, the intercellular space. So intercellular space is what? The space between the cells, right? So these mesophyll cells, they have intercellular space, while they are, because they have compact arrangement, they do not have intercellular space. Okay. Now, if we talk about the chloroplast, so what kind of chloroplast is there in the mesophyll cells? So we know mesophyll cells have what, granule chloroplast, right? Because it has structure will be like this, and they are connected to another, right? So it has granule thylakoids, so it has granule chloroplast. While in the bundle shaped cells, the chloroplasts are dimorphic in nature. So they have dimorphic chloroplast. Now, what does dimorphic means? It means that in the leaves, there can be different type of bundle sheet cells. Suppose in one cell, it has chloroplast that is granule, while in another cell, it has chloroplast that is agranule. Like here, it has just thylakoid. There is no stack of thylakoid that is forming the grana. So dimorphic means have both granule and agranule chloroplast. Okay, granule and agranule chloroplast. Okay, now because they are used for photosynthesis, so let's see what photosystems do they have. So the photosystems, as we know, in the mesophyll cells, it has both PS1 and PS2. But in the case of bundle sheet cells, it has only PS1. Okay, it has only PS1. Now, if it has only PS1 and it has PS1 and PS2, what kind of photophosphorylation will happen in both cases? So the photophosphorylation. So because it has both PS1 and PS2, it will have both cyclic and non-cyclic photophosphorylation, right? But here, in the case of bundle sheet, it has only PS1. Now, if you remember that there was one kind of photophosphorylation in which only PS1 was active. And that photophosphorylation is what? It is cyclic. So in bundle sheet cells, it has only cyclic photophosphorylation. Okay, now if we talk about O2 production, which of these two cells will produce O2? So the O2 production, if oxygen production, it is associated with which photosystem? It is associated with photosystem 2, right? So the oxygen production will take place in the case of mesophyll cells. So here oxygen production will take place. While it has PS1 only, so there will be no oxygen production because it will undergo cyclic photophosphorylation and that does not require water splitting and hence oxygen production, right? And lastly, enzyme, if you talk, ribisco. So in case of C4 plants, okay, not in C3, but in case of C4 plants, the ribisco is absent in mesophyll cells. Okay, it is absent in C4 plants. Okay, in case of not C3 but C4 plants, in mesophyll cells, ribisco is absent. Instead of this, an enzyme known as peptase is present. Okay, so here instead of ribisco, in mesophyll cells, peptase is present. While in the case of bundle sheet cells, it consists of ribisco. So the bundle sheet cells have ribisco here. Okay. So this is the difference between the mesophyll cell and bundle sheet cells in the case of C4 plants. Now, in the next lecture, we will discuss about the advantages and disadvantages of this C4 plant, and then we will move to the C4 cycle. So that's all for today. Thank you.